Good afternoon and welcome to the second day of the General Assembly. I hope the participants and the involvement of all of you will be in the same line as yesterday, with full discussions and sharing ideas about the topics that we are planning to manage today. It will be mainly three topics to be covered today. First one is the sustainability of thinking in the future. It is quite important. It was also a message and advice coming from the reviewer panel last November. We must be focusing sustainability. And the second part will be focusing the future of harmony. And one part of the future of this harmony is, of course, sustainability, but the other one is Harmony Plus and the new projects coming. And these are the three main steps in which we are now, we will discuss. The first point is sustainability. It is really an important topic. Harmony approaches several companies in order to give or to ask for advice coming from these different companies. They are related to business. Most of us, we are not economists, we are just clinicians and scientists, but no anymore business people. And after some discussions, we decided to go to Estradu for doing and for manage this sustainability in future. And it is the reason why today we have invited the CEO from Estradu, Dr. Michael Müller, who will provide some guidelines, some general guidelines about how is the situation that we are now in which is the present point in which in the discussions regarding the possible model, future model for harmony. We perfectly understand and we know that harmony is not really an easy project to be managed. But on the other hand, I think it's really important to have Michael with us and providing us advice about this sustainability. So let's move to the presentation coming from Estradu. Welcome, and after the presentation we will have questions. We will follow the same scheme as yesterday, raise the hand, and if possible to be able to manage all the full debate after the presentation of Dr. Müller.
Good afternoon, ladies and gentlemen. It is our pleasure to be here as a team member of Harmony for the first time in a General Assembly, kind of new kids on the block as we joined the initiative relatively late. And as you can see here, the purpose of the meeting today will be to give all consortium partners an overview and an information about what is the current status quo of the sustainability project, what methodology has been used and what interim results have been achieved, and also inform about next steps and timelines related with the project. Only to remind you the scope of the project, the objective of the sustainability project. I'm sure you, in principle, you are all aware, but the objective was and is to develop a business strategy and a business case for Harmony to be sustainable after the grant period. That means to generate a strategy and a case describing what kind of funding sources Harmony can have, may have, should have after the grant period is over and there is no more granting of funds from the initial initiators of the project. And in order to run the project, it had been structured in 4 phases. We started with an assessment and a mapping of expectations. Assessment means we we made transparent, available data, scope of data, comprehensiveness of data. And of course, that changed over time, because when we started the project, there were almost only half of the amount of data on the platform that are on the platform today. So it's a moving target, obviously. We also mapped expectations from internal stakeholders as well as from external stakeholders, what they would expect from such a platform in order to deliver and in order to what benefit it could create. Then in phase 2, we tried to make transparent what are the cash burning elements, what is the money spent for in the current situation, but also to translate that into considerations. How would money have to be spent after the grant period? Asking what kind of services may sustain and will be provided in the future. We have been assessing the resources on the platform of who is doing what, how many resources are required, what is done internally, what is done externally with vendors and partners. And then over the time, we started building first hypotheses. That was towards the end of phase 2, and then at the beginning of phase 3, building hypotheses, how business models for a sustainability of Harmony after the grant period could look like. That means, and I will come to that in much more detail later on, asking what kind of services, what kind of products, what kind of sources for revenues or fundings could exist after the grant period that would allow Harmony to live on even after the grant. And that phase 3 is actually where we are currently in, and you will see that in more detail in a minute. And in phase 4, which is the last project phase, which is about to come during the next 6 to 8 weeks, we will be defining a business strategy and a business case that will give revenue assumptions on revenues to be generated after the grant, and also on costs that will occur in order to fund the resources needed to generate the revenues. This slide illustrates the project time so far, and what you can easily see, at least the ones of you who have been familiar with the project before today. We started in March, and initially our all plan was to finish the project before the summer break, so by July. And then what nobody of us knew, the COVID pandemic stepped in, and we had to pause the project work. We had to first pause methodology, then change methodology, and work remotely. In the very beginning, meaning in April and May, we first thought that might be over soon, so that we could come back to the initial methodology after a while. But then we realized after 2 months of COVID pandemic that would not be the case. And that was the main reason why we, after intense discussion with the executive committee and the steering committee, decided to change the methodology and go away from smaller physical workshops and more interviews to a survey that we had run then in the months of May and June, an online survey that was addressing all consortium partners and gave the opportunity to everybody to feedback on a huge amount of various different topics. And I will come to that also a bit more in detail later. Next slide, please. But only to give you a brief overview. So in those first 2 phases of the project, we have been involved in many individual interviews with the management and the hosting partners of the platform, with Jesus and Santiago, but also with the secretary on budget and spendings. We have done single interviews, partly at the beginning physical interviews face-to-face, later on then telephone interviews with various of the key partners from the clinical side, but also with one of the key partners from GMV supporting the Harmony work. But also we did a lot of external stakeholders and experts interviews, people who are involved in similar activities or who could serve as experts based on the experience that they have, people from commercial corporations dealing with real-world evidence and publishing scientific information like IQVIA or Elsevier, one of the major publishing houses in the world, representatives from Helmholtz Institute and other institutions, but also a lot of other representatives from other pharmaceutical companies involved either in Harmony or in other platform projects. So some of the members that you see here that have been interviewed are also involved in other IMI projects and have experience from other projects with a similar nature, but in different indications. And what we have also done is anticipating a little bit what are the services, what are the future business models that a platform like Harmony would be engaging in. And based on those thinkings, we have started to create benchmark information on other activities that are comparable to some extent, that are different in other extents. For instance, you see on the very left and in the middle on the top, you see two other IMI projects, one on prostate cancer, the other one a call that is currently running, a major call on 5 solid tumors with various partners involved. On the very left in Pioneer and Pioneer Plus, there is also sustainability consideration ongoing, and in some of the topics they are much more advanced than Harmony is today. In Optima, which is a call that is currently running, that will be started, the project will only be started in 2021. But the difference is that in the new IMI calls, the sustainability as a work package is embedded from the beginning. You remember, all of you have started with Harmony without that sustainability already be embedded in the processes and in the consideration, and it had been, as a project, had been docked on relatively late, the beginning of this year, while in the new projects, as it is so important, it is embedded from the beginning. And there is also from the beginning already in the period of the call to be submitted, there is already discussion with external partners on potential commercialization models for the platform. We have benchmarked the initiatives with other oncology platforms, such as Clinical Path or Flatiron in the US, which are to some extent highly different and cannot be compared, but in other regards are highly comparable to what Harmony does. So there's definitely a lot that you can see and learn from those. We have been speaking and benchmarking initiatives with players like Smart Patient, that is one of the leading developer for digital therapeutics, who are generating currently a lot of real-world evidence that they market. So it's a different and a very innovative model of marketing real-world evidence that is generated from treating patients. And of course, we have included in our benchmark analysis the Multiple Myeloma Research Foundation and Medidata, which is an interesting provider of medical information and data, generating today almost 700 million of revenues from providing evidence and medical data. We have included the Cancer Genome Atlas in looking what they exactly do and provide and how they are funded. And we have included companies, and that is not an exhaustive list. The chart only serves to illustrate you the various let's say, different clusters and archetypes of initiatives that we have benchmarked throughout the project. In the lower right corner, you see a kind of new company, which is Tempus. Currently one of the fastest growing and most innovative platforms for genomic sequencing data in oncology. A company only recently founded with a $5 billion backing for several activities that, and we will see that later on, what they provide is clinical decision support, is real world evidence marketing, is profiling patients for clinical trials, is companion diagnostics matching for stratified medicine on genomic sequenced patients, etc. So a whole lot of activities that they cluster in one platform, and some of that is very relevant and very comparable to what Harmony may do in the future. And it is a fascinating example of how much funding such a topic, such a theme that we are all working on how much funding such a theme can generate. Now, I mentioned that as we had to change the methodology and go away from the physical meetings in April and May that were intended and foreseen in the methodology, that we started that large online survey. And that was a survey addressing different topics of Harmony and the platform work. The first chapter was about the status quo and the general achievements so far. Second was on infrastructure, then about the collaboration with clinical centers and the collaboration with the pharmaceuticals industry. The performance of the different work packages, initial ideas that consortium partners and members would have on what should happen with Harmony after the grant periods. And if something should happen, what would it be? What kind of business models could they think of that Harmony could be involved in? And finally, initial ideas and hypotheses on future corporate structure, governance, and there was a specific section also for the clinical centers only, which dealt with cost of data, circumstances to provide data, nature and freshness of data, etc. That had questions only for the clinicians. And in the following few slides, I want to give you a little bit of an overview of the results. That is, of course, not the detailed report of the survey, but it highlights the most prominent and most important results of the survey, consolidating the opinion of the attendees of the survey. So first, good news you could say is that almost everybody is convinced and was convinced that Harmony should continue to exist after the grant period. 96% of the participants of the survey said that Harmony should go on after the grant. They believed that there is a strong potential for commercialization of services, and that commercialization of services would be the source for funding future activities. And saying commercialization of services does not necessarily mean in a for-profit organization. It could mean, and that was not the nature of the question, it could also mean commercialization of services within a non-for-profit organization that uses services and revenue generation in order to self-fund their activities going forward and not to generate profit for the shareholders of whatever company. However, there were also very clear and manifold signs of significant existing hurdles and shortcomings that would have to be overcome for Harmony if it wanted to exist after the grant period. There was, in general, a high appreciation for the management of Harmony today and the personal commitment of the management within Harmony as for today. While some members also questioned what, with regard to the location, what might be going forward a best location for hosting such a platform. And what also had become obvious in the survey was that most of the participants from various angles and perspectives commented that the achievements so far, that they would lack expectations regarding accrued data, regarding research questions already tackled, regarding regarding results already generated. With regard to the processes, the attendees commented that the process for data acquisition would have to be improved with regard to timing, but also with regard to how and who would contribute to data acquisition. They commented that there was a limited involvement, that there has to be a limited involvement of certain stakeholders when it comes to decision making in order to be more efficient and faster in the future. That of course referred to some specific work packages, not in general. And it also had to be seen in the light with limited veto rights that needed to be there, or that were perceived to be needed in the future when it comes to deciding on research proposals and being unbiased and efficient in decision making. Attendees commented that the process of identifying new centers and recruiting specific patients with specific mutations, etc., would have to be improved and speeded up. And they also commented that the collection of clinical data was not satisfactory so far as for today. Various aspects here. Comments on not enough data, many comments on not fresh enough data, not young enough data, and some subgroup of participants also mentioned that they believed that the compensation for the data was not enough. With regard to the infrastructure, there were comments on a too long process for data harmonization and the TTP process. They also requested better tools for transparency and availability of data on the platform already. Many comments on a professionalization on the website for two different purposes. One, attract more members, and B, to provide better platform communication, or to use the website in order to leverage and enable better communication within Harmony. Some comments on questioning going forward and in a period after grant, questioning external providers for services with very different aspects here, and of course not uniformly on one topic. Many comments dealing with the organization of Harmony per se. A lot of them asking for improved outcomes and efficiency in the work packages, stating that there was a need of a better leadership in some of the work packages, that there were hinders due to the nature of the project, which is the European IMI project that requires a lot of what participants perceived to describe as bureaucracy that would probably have to be overcome after the grant to be better, quicker, more professional, more effective, etc. A better data analytics and therapeutic evaluation that was said to be not satisfactory today, and a very strong commenting on work package 2, with 60% of the answers commenting that work package 2 should be more unbiased with regard to formulating research questions or accepting research proposals, etc. And that also handed over to the request for a better and more effective internal alignment of the partners from the pharmaceuticals industry. Overall, the attendees of the survey requested a more collaborative structure, including not with regard only to this, but including an improved collaboration with the pharmaceuticals industry. And they also mentioned that the current structure of the organization as a project with an executive committee and a steering committee would definitely need to be modified after the grant period in order to run Harmony more like a general management instead of that multi-level and multi-angle complex organization that it has today. When we asked for what were ideas, what were hypotheses on what Harmony should be doing after the grant period and which business models could be used in order to generate revenues and funding, 84% of the respondents mentioned and were convinced that Harmony should go for commercial business models in order to fund activities. And that was a long list of activities, but the most prominent ones, the ones that were mentioned in almost all of the answers of the survey, were building AI tools focusing on hematology, undertaking dedicated research questions for either institutions or pharmaceutical companies, designing in silico virtual clinical control arms out of the data, possibly create a clinical decision support tool. I've been writing here a la Flatiron, not comparing Harmony with Flatiron, but using Flatiron to explain what a clinical decision tool could be good for and how it could look like. Providing a clinical trial finder for patients with specific mutations for each and every center across Europe treating patients, and the commercialization of the real-world evidence that is, without any doubt, created on the platform in a very huge extent, and that real-world evidence could be commercialized either with regard to research questions for the pharmaceuticals industry for marketing purposes, but also for governmental purposes and health technology assessments. So very different aspects here, but all relying on the real-world evidence generated on the platform. So let's say summarizing on the survey, and as I said, it's just a brief overview of the manifold results that the survey has given and the various subgroup analysis that we had done on the survey, asking what answers come from what subgroup of attendees, etc., and to see the differences here. But as a summary, the phase 1 and phase 2 of the project have given very clear guidance on achievements and existing shortcomings. And they have also indicated that there is a clear need for change in order to continue after the grant period. But they have also given a clear proof that there is a strong expectation that Harmony would continue to exist after the grant period, and not only a conviction, but also a willingness of almost all consortium partners. And interviews and survey have started to give a bunch of initial ideas and components and hypotheses of how future revenue generating business models for Harmony could look like, and how operations could look like, what structure could be used further on, and what governance might be required. But of course, and that indicates that we are only not even at the end of phase 3 of the project overview that I have shown at the beginning of the presentation here. Of course, there are quite some questions remaining now in order to to complete the project. And those questions now are, number one, what exactly is it that we will do? We have a lot of ideas, we have very concrete ideas and some vague ideas, but what exactly is it that we would like to do? Where is the focus of Harmony? What business models will we engage in? What revenues will that generate? And who will be the clients for those services? And then after that has been answered, the next subset of questions is, how do we do it? With what resources, which infrastructure and technology, what cost will occur in order to do it? Because future setting is different from existing settings. So it would be not correct only to extend the current existing annual budget for Harmony in order to come to a future budget. But budget depends a lot on question number one, what is it exactly that we will do in the future? And then third group of questions, in what kind of organization will we do it? Will that continue to be a partnership, a consortium? Will it be a foundation? Will it be a commercial company, etc.? And other questions around that topic. So in order now to collect all the pieces that we have and to bundle and focus it into those group of questions now, we have decided that towards the end of the month, we will start with two important workshops. The first workshop will be on October 28th, and I believe many of you have already seen the invitation for that sent out by Santiago. Those workshops will be, we hope that they will be, and we plan for that, in part physical workshops. So there is currently still the planning that at least a subgroup of workshop participants would meet in the Madrid area and come together for those workshops, while several other participants would join remotely. And the first day will serve to answer the first subgroup of questions. What is exactly that we will be doing? What business models will we engage in? How will we generate revenues? How much revenues will that be? And as the question has popped up in one of the previous internal discussions that we had, I've just wanted to show you a few examples of how the detailed questions that we are going to discuss on the 28th will look like. For instance, one business model that has been mentioned very often, and that might be a very obvious business model for Harmony in the future, would be to provide clinical trial, in silico clinical trial arms, instead of actively including new patients, enrolling new patients, using existing data to build an in silico arm for a clinical trial. So if that is a business model that you want to embark in, then the question would be, what kind of clinical trials will we be seeing in the next years to be addressed to the platform? Does Harmony have enough data to answer those questions? So to build a clinical trial arm with the data. Is that enough data? Is it good enough data? Is the structure of the data suited in order to build such a clinical trial arm? Do we have enough centers that will be needed? You all know that for clinical trials, it is not only relevant to involve a certain number of patients that are suited for the clinical trial, but also to enroll a certain number of clinical centers for various reasons. You want to enroll several different regional centers, clinical centers in different regions, in different countries, etc. And a sub-question will be, what will be the cost to acquire the data for that continuously? Because it is obvious that if you want to to build clinical trial arms, the data has to be always fresh. You cannot build a clinical trial arm in oncology with 10-year-old data. That does not make sense at all. So what will be the cost always to have up-to-date and fresh data on the platform? And then a significant question is, what would the pharmaceuticals industry be willing to pay for such clinical trial arm composition on the platform? So what revenues would that business model then generate? And a question that is also evident and not trivial is, who will commercialize those services? Because let's suppose it will happen. Let's suppose there is enough data. Let's suppose the pharmaceuticals industry would want to buy the data for such a clinical trial arm. Harmony would only be in one subset of indications in the medical world. So the same questions arises in other oncology, arises in cardiology, arises in all kinds of disciplines. So of course, the commercialization of such services might be depending on scale effects and might be better in other hands, in the hands of a company that is providing such services across the indications. So one could imagine that we generate the data, but we not on Harmony do the commercialization on our own, but would run into an alliance with a company that typically runs clinical trials. So companies like Quintiles or others, several around. And next question, would we do dedicated research questions answering? What type of research questions would that be? What level of specificity can we deal with? Are we handing out the data, or are we only handing out the information? And are we collecting the information from the data on our own, which would then in return require that we have an organization of data scientists that is getting the information out of the data? And that has to do not only with the question, what do we want to do, but also with the question of data protection and data laws in Europe, which are different than in the US, and so on. So I don't want to get even further. You will all get those slides and the presentation of today that illustrates for you later on as a read what could be questions in selling real-world evidence, what could be questions in providing a clinical decision support tool. Is that relevant for Europe? Does Europe need a clinical decision support tool in hematology? I am pretty convinced that many of you attending today would not need a clinical decision support tool. On the other hand, many of the clinicians not attending today in Europe would probably benefit a lot from a clinical decision support tool that would give clear guidance on what to do with the patient, and maybe also give information in which clinical trial to enroll the patient, etc. So those are the questions that have to be answered on the first workshop on the 28th. And then, as that is the second most important, on day 2, on the 29th, once we have maybe not finally completed, but a lot narrowed down from day 1, the information, what is it that we want to do? We can then conclude on the question, in which setting? What will be the corporate structure for that? Will it continue to be the partnership as it is today, or will it look different? And as that is a highly complex question, that second workshop is currently internally prepared by rounds of internal discussions orchestrated by Santiago and Jesus with the executive committee, with the steering committee, etc., in order to have already guiding ideas and thoughts for that workshop on the 29th. And then after those 2-day workshops, at a non-defined point in time and with a yet non-defined group of participants, we will then be in a position to answer the remaining question, which is much easier than the first 2 subsets of questions, the questions on what resources, what cost, what FTE behind, what technology, which services, or what kind of work done internally and what with external vendors. And that is something that we can answer then after the first 2 workshops have been completed. You can all imagine, looking at those questions that you have just seen, how difficult that is in a world of pandemic today. Those questions, they would be difficult to be answered in a group of workshop participants sitting for a full day around one table. And it is, of course, a lot more difficult to discuss those questions with participants only participating remotely. So what we will do, we will try to prepare those workshops at the best, using innovative workshop methodology like WooClap, for instance, which will allow in the workshop to use flip chart and brown paper discussions and methodologies as if we were physically in one room. We have, well, fortunately, unfortunately, during the last 5 months, we had to be in a position to collect a lot of experience, because all our work with our other clients in our consulting projects, unfortunately, in the last 5 months, did not happen in physical meetings, but took place in remote meetings. So we have been engaging a lot in such workshop methodologies. That comes back to the remaining time acts, showing that phase 3 will now conclude in the 2 already defined workshops on 28 and 29, and then in a remaining subset of little workshops for the remaining questions in the days after. And then we expect to be in a position to finalize phase 4 of the project within another 4 weeks or so, in order to hand over sustainability strategy and implementation recommendations. Yeah, and that only illustrates an already pre-discussion that we had in the core group of the project, thinking of who could probably contribute a lot to the different question settings, to the different workshops and subsets of questions. But of course, this is not a final list. What is clear, though, is that the workshops will only be possible with a limited number of participants. You cannot run those workshops with 50 participants, so we will probably have to limit the workshops on day 1 and 2 to approximately 10, 12, 15 participants, depending a bit of the nature of the participants, so that it still remains handleable with the remote technology. But I know that Santiago has already sent out the invites. There will be more concrete invites for the ones who commit to participate to the workshop, with pre-reads to the workshops and explanations for methodology, etc. And we may also, only as a head up, we may also reconsider the timing that Santiago has put in the email, because both workshops so far in the timing already addressed were planned to start in the morning, Central European time. However, if it turns out that there is a need and a benefit to include a significant number of overseas participants into the workshops, then we would probably reschedule and start the workshops later during the day in Europe, so that US or other abroad participants have a chance, or have a better chance to join without getting up 1 o'clock in the night. Thank you very much for your attention and for listening. And now it's open for questions.
So thank you, Dr. Miller. Really this comprehensive story about sustainability and the results from the survey. We have 4 questions in the chat. Renate Schulz-Rath made a question: are the results that you are currently showing from the majority of the participants, or only the opinion of one or two people?
No, what we have shown in the overview is clearly not the answers of one or two people, but we tried to summarize in the set of charts what is the majority opinion coming from the platform. Of course, you can imagine if you have seen the survey, there were a lot of open questions in the survey. So everybody is phrasing it slightly different, but all the topics that we have mentioned are not single opinions. We have completely excluded single opinions here and only summarized what is majority opinion or has been a clear sign throughout all the answers that we have received.
Sophie Wintridge asked a question regarding the work package 2. It was you made a comment that someone told that some people think that they are biased project, and she told more unbiased in that way. In what way? What kind of bias was observed?
Well, the question that is addressed here is whether a work package that is deciding on research questions should be completely free of particular interests with regard to partners from the pharmaceuticals industry. And that referred to a more general topic to it, saying that if we are deciding on scientific questions, those answers or the decision should be completely free of the pharmaceuticals industry opinion, which of course is only a subgroup of the total pharmaceuticals industry because only a couple of companies are attending. And in more particular, it was addressing the topic that certain aspects of clinical decision or clinical questions are kind of excluded, like head-to-head comparison, etc. And the third element that was addressed here is that foreseeing the possibility to sell real-world evidence and innovation and information generated on the platform to governmental bodies, for governmental authorities, for health technology assessment. The prerequisite for those governmental bodies might be that the data was generated completely unbiased from the pharmaceuticals industry. That were, let's say, directions of where the answers came from.
Okay, Valeria Santini, Professor Santini from Florence University, made a comment, and this is, I think, is also a very important comment. Interesting to her talking about business models to further support Harmony, but please do remember that universities have ethical and conflict of interest limitation in this sense. How we will manage?
Yes, a very relevant question. You may have noticed that a couple of minutes ago I said one question, for instance, will be, do we provide access to the data, work with the data, or do we provide information? And in a European context, we are providing information generated by the data. So we will have to find means that are compliant with our European understanding of data protection laws, of scientific ethics, etc., to take a research question up, whether that is an ethical research question, to provide the answer out of the data. That could be by opening an anonymous data room, which will not allow the client of the data to misuse the data, but it could also mean that Harmony itself has a group of research data scientists that are providing the answer. And that is clearly understood, and that is something that is addressed currently in each and every IMI project, that any kind of commercialization, of course, has to be compliant with European science and data protection environmental aspects.
Okay, thank you, Dr. Miller. Professor Andrea Manca from York University required something about the real-world evidence data. is to provide real-world evidence data to government or HTA bodies. We have to demonstrate their values for HTA-like decision-making, how we can manage this.
This is a question that I have to give back because this is a question to the clinicians, a question that would be addressed in workshop number 1. What is the real world evidence that we can draw from the existing data that we have? What benefit would it have? In which aspects could we give benefit to current questions of health technology assessment that we are aware of? So this is a question that the clinicians will be in a position to answer much better in a guided manner, but much better than I would be.
Okay.
It is, maybe to be more concrete, it is maybe just to add, Jesus, it is obvious that real world evidence as it is existing on the platform, because the platform is one of the very limited that exist in the world, that comprising all kind of different data for one patient and for groups of patients. Nowhere else you find the information on pharmaceutical treatments, information on outcome, information on diagnostics and patient characteristics. Each subset of data today is lying somewhere, and there are commercial models for each of the subsets of questions, but very few people are in a position to combine all that information and to generate evidence out of it. And it is obvious that you can get better evidence if you have all that information at hand, as if you only look, for instance, into IMS data, where you have limited evidence, or if you look into old meta-analysis of trials under very different conditions, instead of looking into a subset of clearly defined fresh data. That is the idea behind it.
Okay. So I also have, we also receive a message in the chat. You can also read. I will read for those who don't have access to the chat because they are connected by phone. It's a message from Kathleen Hewitt. Kathleen, together with Bill Wood, are attending this workshop today. Join us. They are members responsible from the ASH Data Hub. And Kathleen told us early this year, ASH Research Collaborative completed his business model project, and the business plan was launched this summer. We are happy to participate on the workshop if helpful, designing a business to address the needs of various stakeholders, commercial and non-commercial. It's not easy, of course, but can be done. Congratulations on your progress so far. Thank you so much, Kathleen. I think you are welcome. We were in touch, and I will use also this forum to tell you that maybe we can have a TC next week, or in the next 10 days, we can organize this TC that was to continue with these links between Harmony and ASH Data Hub regarding not only myeloma, but also these other topics that we are managing now. Thank you, Kathleen. Thank you, Bill, also for attending the meeting today. I don't know if there is any other questions. We are just on time. Maybe we can have a last question if someone want to raise the hand or make a kind of...
Did you address the question of Valeria Santini, Jesus?
Yes, it was already addressed. Yes, it was.
No, maybe one comment on my side, Jesus.
Okay, John Butler from Bayer.
Yes, I just want to encourage everyone, in particularly our academic physicians, our so-called KOLs, to make an effort and participate in the workshops planned for next week. I think, sorry, next week, no, the week after next. I think this might all sound a bit confusing, but at the end of the day, the most important thing that needs to be determined is, where do you want to take this consortium? Where do you want to take this endeavor? Do you want to end it? Apparently not. There was an overwhelming majority advocating for continuation. The question is how. And if it's going to fulfill the original vision one day of registering, if not everyone, but at least a very substantial fraction of all European patients in hematological malignancies, to collect that data, longitudinal data, in one big research resource. Then the second question is how can we make that work, and how can we generate the necessary funds? And I'm very optimistic that there will be no unsurmountable ethical questions here, because let's not kid ourselves. We're not going to become rich here, personally rich. We're talking about allowing this resource, this research resource, to survive the end of the AMI program and to thrive in the future. So it will be extremely important for the KOLs to participate and give their opinion and their willingness to commit in the very first phase of these workshop in 2 weeks. Oh, thank you very much.
Thank you, John. I fully agree with you, and I fully agree with this point coming from one of the FPM members from the steering committee. So I will fully support this, let's say, this open question. This is a challenge for all of us, for all the clinicians, for all the key opinion leaders, to really provide their ideas about how they foresee the future of Harmony, and which is all the potentiality that we will have in the consortium. And I think it is today is the day in which we have to encourage this participation. We will invite from the Harmony office. I am quite sure that not everybody or everyone is opening all the mails that they are receiving from the Harmony office. And I will take care, and I will send a personal mail to all of key opinion leaders in order to get as much involvement as possible in these meetings, in these workshops that will provide the best ideas about the future in Harmony. So thank you so much for actively. I don't know if Dr. Müller want to add some further last comment.
Yeah, maybe just two concluding remarks here. First, addressing to what John Butler just said. I think that is a very important message, because I believe, and I believe that you believe, that what you do in Harmony is the only right thing to do in order to make research better and to make treatment of patients better. You all know that single clinical trials and single center aspects, especially in diseases which only have few patients in one center, that is the only correct thing to do. Pooling data, getting the information across a huge number of patients, getting all kind of information in a comparable, what means standardized, in a comparable manner. Data from trial 1 comparable with data from trial 2, from center 1 to center 2. It is, you all are convinced that this is the only right thing to do. So if you're all convinced, think of what John said. There are only two possibilities after the grant period. Possibility number 1, Harmony is over. And I surely believe, and the survey has shown, none of you wants that. All of you want that Harmony is sustainable and continues to exist because it is good. So the second option is, if option 1 is not a valid option, the second option is to conclude and to find a structure and a set of services and a mode to generate revenues for Harmony to exist after the grant period. And we all know that this is not easy. We all know that also with the project, we have been tackling very sensitive questions, very different perspectives and interests of the people contributing to this fantastic work. But let me remind you, we are now towards the end of the project. The 2 workshops are extremely important, and we would be grateful for each and every strong opinion participating in the workshop and making those 2 workshops on the 28th and the 29th a success. That was comment number one, and comment number two is only to express my thanks for all of you contributing so far with feedback, with interviews, with open opinion, with a lot of criticism on certain remarks, etc. That is great, and this is exactly what we need in a consortium like that. Thank you very much. We look forward to supporting you towards the end.
Thank you, Michael, and thank you so much. And of course, Harmony is not an easy task from the very beginning. So, and we have to continue to be and to survive in this non-easy task. But this is something that we manage usually. Most of us are clinicians taking difficult decisions all the day by day, with difficult patients in order to improve their quality and quantity of life. So it is something in which, okay, it is our routine in the day-by-day scheme. Thank you so much, Dr. Miller. And we are now going to following the agenda. And the next topic will be the future of Harmony. And for this topic, we have invited two members of the executive committee, Guillermo Sanz, professor in hematology at the University of Valencia in Hospital La Fe, and Dalal from Celgene, who will take care of the future, or the future, how about the future regarding Harmony Plus in the next coming projects.
Okay, so thank you. Jesus, yes, Jan Ludwig, yes, for Celgene BMS, and I'm happy to be calling for IFP with Federico on the help of Alneta, also from Celgene Bristol Myers Squibb, a new member as IFPA member, because Celgene is part of Bristol Myers Squibb now. So, as mentioned by my colleagues before, Harmony is about data. It's about many other things. It's about collaborative mindset. It's an IMI project for with public and private partnerships. So it's a two foundation of IMI. It is this collaboration between private companies and the public organization and patients. So it is the beauty of this consortium. It's that we have all the stakeholders around the drug development in hematology involved in this project. So you see in this table the data that we have so far and what we plan to have. So we are clearly not on the target. We need to achieve more data, to have more data in the platform for sure. If you make the sum of the line on the bottom of the table, you will have 65,000 cases as a target now, based on commitment. Our goal at the end of the project is to have 100,000 cases in the database. So we have a lot of work to be done on the public side, but also on private side. It's really an important commitment, and we need to achieve that. So I encourage my colleagues who are present here and representing the pharma that are contributing in kind and cash in this kind of big projects to really make the maximum of effort to authorize the capacity to share data. It's a very complex action at the level of the pharma for plenty of reasons. As you know, after IMI, we have IHI, probably other kind of collaboration, where we are speaking already about IP right and plenty of important topic, important impact challenge that we have to face in order to release this kind of data. It's we are convinced, at least Celgene, Bristol Myers Squibb, I'm sure all my colleagues who are on the line, future it's on collaboration between private and public organization. And the goal is really to share data now. It will be the future in any case, notably after this COVID-19 crisis. So yes, it's really an important topic, and I hope that we will be able to do it in the next years. So currently we are on the platform from ESPA, Novartis database, Takeda database, and upcoming Celgene, Bristol Myers Squibb data, Janssen, and Pfizer. As I mentioned before, it's not enough. We need to have more partners involved. I think it's also about clarity about the future. It's really important to be sure that I think now we think also about the future perspective, because we have to reassure about plenty of aspects regarding the use of this data. And I think I want to raise here an important point. My colleague John mentioned the importance of the participation of experts in KOL in this next workshop. I think it's also important to not hesitate to ask questions. It's a very complex platform. It's innovative for the collaborative mindset, but it's also innovative in a technical point of view. So I think it's important to remind that it's not a repository, so a sort of C drive where we have a list of studies where you could pick up and make some comparison between studies. It's a platform where we are mixing the data, aggregating the data in order to get results based on project research that we are discussing in common. And the goal, in my opinion, but I hope that I'm sharing that with my colleagues, that we are in fact committed to achieve some project research where we are in agreement. Because obviously, all the partners could have different interests, agenda, all without exception, public and private. And but we have, and you know more than me, and I'm speaking to the KOP leader now, there are plenty of unknown in all these, where we could all focus and where we could find a common interest. So it's really important to have that in mind, that to not hesitate to ask questions to the different experts that we have in Harmony, technical point of view, for example, how the data are used, how they could be used. It will be also useful to release some nice idea about project research, for example. What the demonstration made by Professor Bullinger is very important regarding the result that we saw. And I think there are plenty of opportunities. And I hope also that the pharma, when they will provide some project research and ideas around that, because it's important for everybody. So the future in terms of fact and regarding the meeting where we are presenting results on our project will be of September. We have the IESA Congress and the meeting for IMS, but it was done in July. So, as you know, ASH is coming. It will be also virtual. Harmony will be presenting. IPSOR also will come, which is an important meeting for around the globe. And we will have also, it's an important topic, and I think Regis will speak about that also, the General Assembly next year, according to what we see in the pandemic situation. And I hope that we will have the occasion between this now and this important day to get some results.
Thank you, Jan. I will continue with this presentation on the future of Harmony, and this is a clear example of one of the main achievements of Harmony so far. We have been able to create a big data platform for hematological malignancies, in which the data that are included are harmonized in the OMOP common data model. And this is critical for a systematic and standardized analysis, and will allow us not only to perform our own internal analysis and research projects, but also to connect with other organizations. And as you know, we are currently establishing different kinds of cooperation with our colleagues at ASH, others in the Southern Asia and Australia, and in China. And this is very relevant, because our platform is not only allowing this kind of analysis, but it has shown that it is a safe platform that guarantees the anonymization of the data of the patients that are included in the database. As many of you who attended the Harmony session number 1 of Harmony yesterday evening. There are now several different projects running for the different hematological malignancies. And we have also feel that Harmony could not let pass the opportunity to start a specific project on the effect on the outcomes of patients with hematological malignancies with the COVID-19. We need a new research project for all the different pillars of disorders that are already included in Harmony. And I would like to stress that this research project can come not only for partners, but also from associate members. And I would like again to highlight that I would be very delighted to see that our colleagues in the pharma companies could be able to propose a specific research projects for the different disorders. Again, we are trying in some way to give our own view of the incidents and also the consequences of the COVID-19 pandemic for patients with hematological malignancies. And this afternoon, we will have the pleasure to have 2 different talks, one by Professor Tiziano Babui on the impact of COVID-19 in patients with myeloproliferative neoplasm, another by Professors Pagano and Connelly, that is an open collaboration of the Working Committee on Infections in Hematology of the European Hematology Association, in which we are starting to create a collaborative effort that I think will be beneficial, not only for hematologists, but for the whole society. For these purposes, we have built a case report form for COVID-19 patients with hematological malignancies that is available under demand. And I know, because I have received several comments and criticisms for many partners of Harmony, that this, let's say, CRF is very complicated in the sense that it includes a very huge number of different variables. And it's true, it includes demographical variables and previous medical history of the patient, data regarding the specific hematological malignancy, data on the different signs and symptoms of the COVID-19 in this set of patients, data on the different treatments and the outcome of the patients who receive a specific treatment. Also, data regarding the COVID-19 potential complications, not only short, but also those occurring in the long term. And we have included a very large number of variables trying to find biomarkers of the severity of the disease and of the outcome, including different imaging and analytic tests, especially different blood results. Just one opening slide for the Harmony Plus project that will be given a specific presentation after this one. We think that in Harmony Plus, we needed to extend the Harmony previous hematological malignancies to all hematological malignancies. And that is the way why CML and the other myeloproliferative neoplasms are included, as is other lymphoproliferative disorders not previously in Harmony, like Hodgkin disease and Waldenström macroglobulinemia and other rare, very rare hematological malignancies. We want in Harmony Plus to expand our focus to be able to achieve the collection of data coming from registries and trying to obtain real-world evidence data, especially in outcomes. Again, we are going to involve different European cooperative groups, hospitals, and other data providers not previously present in Harmony. We would like, as a very relevant goal, to be qualified by the European Medicines Agency as a platform that could be used for building new clinical trials and being useful for all partners, both academic or from the industry, to realize new designs of different clinical trials. We are also collaborating in this new project with different IMI projects like Eden Pioneer or eTransfit and many more. And especially I would like to stress that from the very beginning of Harmony, our collaboration with these big data projects of the IMI has been particularly relevant. And I would like to stress the contribution that the collaboration between Harmony Plus and Eden would suppose for the European Union community. And finally, as I said before, we need to enlarge our partnership with other institutions, cooperative groups, and regulators from different countries outside Europe, like the American Society of Hematology, the Australian Cooperative Group, the Korean Society of Hematology, and all other associations that could provide information in the same hematological disorders that are our goal in Harmony and Harmony Plus. I think this is the last slide. Thank you very much for your attention.
Thank you, Guillermo. Thank you, Jan, for these perspectives about future and Harmony and Harmony Plus. And continuing with this, let's say, with this new project, with this new idea, which is a complementary idea of Harmony, Fred Calado and I will, let's say, explain a little bit the Harmony Plus project. For those who really want to enlarge their knowledge in the project, I think the PMOs made a really very nice video presentation, a PowerPoint plus video. Let's go to provide some bullets about Harmony Plus project. It is a duration of 3 years. We started in this month, in October. So we are planning to finish in 2023. It will be around 1 year overlapping with Harmony, maybe more, because we are planning to enlarge Harmony 1 more year. And the budget of Harmony Plus will be almost 12 million. From those, 6.7 million will be direct cash provided by IMI funding. In Harmony Plus, we, from the very beginning, we try to reduce the governance, reduce the architecture of the consortium. And in that way, we made we work in 2 directions. First of all, trying to reduce the number of the work package. And now we are going to be 6 work packages in Harmony Plus, because in some way we fuse work package 3, 4, and 5. And it is because all the work performed by work package 3 in Harmony is used also in Harmony Plus, and it will be more or less the same schedule, the same idea. And also we have reduced the number of partners, both from the public side and from the private side. So this is the general structure. As usual in these schemes, they are really, for me, at least for my understanding, they are really complicated, more complicated to be explained in just 1 minute, but you will have the slides and you can have a look on this. Okay, what is the interaction between Harmony and Harmony Plus? So we have collected and we have performed so many things in Harmony that can be used also by Harmony Plus, that it could be really a very good, very nice opportunity to get all the standard operational procedures for submitting and assess research proposals, for the anonymization procedure, for assessing data quality and completeness. Yesterday we were talking, at least in our ALL working group, how we can manage these, that sometimes is really a work really impressive, but now we have reduced all the bureaucracy, but we have to keep in mind that this is something that we have to retain, because we have to follow the rules, we have to be in accordance with the European law, and that's imply in some way that if there is a data transfer, we must sign this data transfer by contract at any time. So we have to provide the informed consent for prospective data, and we will manage all the data in this data harmonization performed by OMOP. In that way, all the data will have the same characteristics. And I think Fred now can explain better than me the main goals that we are now in mind for this Harmony Plus project. Fred, please, go ahead.
Good afternoon, everyone. I'm pretty sure I will not be able to explain better than Jesus, but I will try nonetheless to explain a little bit the idea. Again, building on what I mentioned yesterday when we started the session, the main goals are multifold in terms of the scope. On the scientific dimension, of course, we are including additional hematological malignancies. Harmony Plus now enables Harmony as an umbrella project to actually cover all hematological malignancies rather than the 7 original therapy areas. In addition to that, there's a huge interest to focus the sourcing of data in procurement of data really internally within the network, within our network, in us getting deep omic data in order to link to the clinical data that is coming from either the studies, the clinical studies, trials, or non-interventional studies. Very importantly, and this has been referred to a few times yesterday and today as well, knowing the growing importance of existing data in regulatory interactions, and especially in those that involve the approval of new medicines through clinical trials that for some reason or another require to be enhanced by existing clinical data from existing trials. Harmony Plus aims to become a source of this kind of information to support these regulatory decisions. And definitely this can and should be done with close collaboration with EMA specifically, of course, in Europe, but also very much open to make good use of the data that will be curated within our platform with other regulatory authorities around the world, as to enable innovative treatments to be approved faster with the existing data. A third category, of course, related to the first one, to the last one, I mean, is on the building of additional modules that will enable these decisions to be made. So not only we want to support data from in the decisions of regulators through the use of data that we accumulate in our platform, but also there's other important decisions related to reimbursement and other health stakeholders that can greatly benefit from the data that we are trying to accumulate in a collaborative way. And then finally, and very importantly, again, I did mention this in passing yesterday, the fact that Harmony is not only a collaborative project within the consortium, but also collaborative with other initiatives in the scientific realm. So definitely we are very much interested to explore synergies with other initiatives. A notable one is the ASH Data Hub that is starting to focus on multiple myeloma in the US, and we are very certain that this will be a fruitful collaboration, as ASH, will certainly in the future also expand their scope to cover other therapy areas that are also covered by Harmony. I'm taking a leap here, but I think this is probably what will happen in the future. And it is with the great pleasure that we are already seeing the usefulness of Harmony and Harmony Plus, even though, of course, Harmony Plus has only been officially around since the beginning of this month. Of course, knowing that this project will come to life in October, a lot of our scientific collaborators have already thought about how we could make best use of this project. Also knowing that the project will be limited in time, will be much shorter than Harmony is, then we definitely want the very good brains around this network to start thinking about what we can do under Harmony Plus. And a very important and a very interesting example is the project that I think we'll hear definitely in just maybe a few minutes from Professor Tiziano Barbui on exploring the impact of COVID-19 on outcomes in myeloproliferative neoplasms. Definitely a very important topic, given, of course, the very special moment that we're living, all of us throughout the world with COVID-19, and very important to understand the impact of this new and very poorly understood condition in patients with MPN. And it gives me special pleasure, given that, of course, I come from Novartis, that also Novartis has also just submitted to Harmony Plus the first Novartis-driven proposal, scientific proposal. It is a little bit different from all other proposals that we've been seeing. It actually is more of a validation study on first preliminary findings from actually data coming from the other side of the world in the US. So the context here is that there were some very interesting clinical insights that were generated with data from Optum, which is a fairly well-known source of data in the US, not oncology specific, but of course with also clinical information from oncology patients. And the idea here is to validate the findings of that initial study with the data that hopefully the consortium, the Harmony Plus consortium, will be able to collect about PV patients. And I'm very, very happy that we were able to secure one of the investigators, Santiago Moreno, to speak a little bit about the proposed project also in a few minutes. Yes, and these are just two initiatives out of hopefully will be a huge volume of projects coming to in our way for Harmony Plus. Definitely, I think, and Jesus made a few comments already on how we try to streamline the organization of Harmony Plus in light of the building blocks that we've achieved with Harmony. Definitely, I think there's always going to be the need for critical mass in each individual therapy areas, and definitely, of course, we will leverage the infrastructure and the governance that has been already put together by Harmony to sort of take these ideas to fruition in terms of research question to results faster than ever, and hopefully we'll be able to prove that actually Harmony Plus can be and will surely be a very cost-effective investment by the IMI office in using already a well-established consortium and platform like Harmony to actually be able, with a marginal investment of cash and in-kind contributions, to actually generate more science that impacts patients with other hematological conditions beyond the 7 that the Harmony consortium originally targeted. Back to you, Jesus.
Hello, thank you, Fred. Now, well, really, for those who really know Harmony, to understand the general structure of Harmony Plus is not so difficult. So we have added, please come back one slide. Okay, thank you. We have added the disease, the LAC disease in the previous consortium, myeloproliferative disorders and CML, Hodgkin lymphoma, and we have grouped the other rare hematological malignancies. But from the very beginning, we have been an open consortium with open mind. So this year, for instance, with the COVID-19, we have to adapt also the consortium to the COVID-19 pandemic. So it is something that is not included in Harmony Plus structure, but in fact is there also. So it is something that we have to manage. We are at the operational level. For instance, as yesterday we met in 6 different rooms. In future, we will need at least 8 or 9 different rooms to meet each other and just to be focused disease by disease, trying to build up the best project as possible. But the general idea remains similar to the previous one. So, well, I only have to tell all of you, thank you for attending the Harmony Plus. Thank you for supporting Harmony Plus project. We will be more than happy if you will require to be associated member with the same rules like in Harmony project. It will be really important to share your data in order to better manage the patients. And also we really need your research proposal. Fred already talked about 2 projects in myeloproliferative disorders. Personally, I'm more than happy that at the very beginning of Harmony Plus, we started with 2 projects. It is really important because it is something that made some difficulties sometimes to really produce a great idea. Now we have these two projects, but quite sure that in CML, in Hodgkin lymphoma, and in the other diseases, it will be also projects. We also have this initiative coming with the working cooperative group by Professor Pagano regarding COVID-19 that we will see later on. So it is really very important to present your own research project proposal, and really welcome to Harmony Plus. Now we are two, Harmony and Harmony Plus, but in fact we are just one idea, one common idea, and the common idea is to really improve the patient management with these hematological malignancies. We are a little bit constrained about the time. Maybe we can move to the next session, and later on after the session, we can have an open forum for discussion in case that you will have questions regarding Harmony Plus, future perspective, or even the project that we will see now. Now, Professor Lars Bullinger, Dr. Ali Farsad will chair the next part, and will chair the 5 projects coming to the Harmony and Harmony Plus. Lars Farsad.
Yeah, thank you very much. So I will start. And we have a small change in program. As Gert Osten-Kopperle will have to leave at 5 o'clock, we will have his presentation first. And I'm really excited about the 5 new projects which we are presenting in the afternoon. I think they will be really good to have in Harmony, and they will advance the project quite a bit. And with that regard, I'm really happy to announce the first speaker, Gert Osten-Kopperle. I think most of you know him, a young hematology professor from Amsterdam, who is chairing the HOVON and one of the lead experts on AML research in Europe, and with a special focus on minimal or measurable residual disease. And this will be the topic of his talk. Gert, the stage is yours.
Yeah, thank you, Lars. Well, thank you so much for inviting me to present a new project in acute myeloid leukemia on measurable residual disease. And it's really a pleasure to be in this virtual meeting, although I really preferred the last meeting in Florence. I have good remembrance to that, sitting on a terrace in Florence, drinking a glass of wine with Hartmut Deuner and Bob Leuenberg. So it's a pity that it's not possible this year. Hopefully next year we will meet in person. So this is to appreciate that currently with chemotherapy, we cure around 40% of the patients below the age of 65. High complete remission rates are achieved, but we are facing relapses in over 50% of the patients. That's mainly due to the persistence of leukemia stem cell progenitor cells that initiate the leukemia. And although we morphologically cannot detect them, we see the presence of those cells in the relapsing AML. Many methods to measure residual disease, and the oldest one is, as I mentioned, the morphology, and that's a very insensitive method. We have now much more sensitive methods to measure residual cells. And one is based on quantitative PCR, mainly possible in a restricted number of acute myeloid leukemias, and currently only the NPM1 mutated and the core binding factor mutated AMLs can be monitored in this way. And it's a very sensitive way, much more sensitive than the other method, the flow cytometry, in which we detect leukemia-associated immunophenotype with a lower sensitivity, but it's applicable in a larger number of the patients. And there are now new emerging technologies to assess MRD. Next slide. Like next generation sequencing and digital PCR. And I will allude a little bit on that further on. So when we find measurable residual disease, then it's the question, what can we do with it? First of all, we can assess the response much better than relying on morphology. And it's now included in the ELN 2017 recommendations. We now have a CR MRD negative outcome. It's possible to refine the predictive of outcome and risk stratification. We can inform our treatment decisions. So should we continue with chemotherapy, or should we apply an allogeneic stem cell transplantation in certain situations? We have the possibility to early identify a relapse and the possibility to intervene earlier. And last but certainly not least would give it us the possibility that it could serve as a kind of surrogate end point for survival. And if that's accepted, then we have an early endpoint that could speed up the approval of all kinds of new drugs. And as you know, there are really a lot of new drugs currently available for the treatment of acute myeloid leukemia. Waiting for the survival endpoints takes a long time, even in the case of an acute myeloid leukemia. And there are a lot of single institute or single trial group studies done showing the association and the prognostic value of MRD status after chemotherapy. And the left panel, you see the quantitative PCR of NPM1. It's a landmark manuscript of Ivy a couple of years ago published, and you see a nice discrimination between the MRD negative and MRD positive population. And on the right hand, you see an example of a study that made use of the immunophenotypic assessment of MRD. Also here, there's a nice separation when you use this cutoff level. And also NGS has been applied in the monitoring of MRD in clinical studies. There are now a couple of examples, and I show here one. And now, and this is really timely, very recently a huge meta-analysis has been performed by the MD Anderson group, published very recently in JAMA Oncology. And they did a literature search of all studies that included MRD and AML in the title. And it was a PubMed search and an Embase Medline search. And I will not go to all the details of this study, but if you can see at the bottom of the slide, 81 articles were accepted for this meta-analysis, and that included 11,151 patients. So this is huge meta-analysis, and the outcome of this analysis are important. So next slide, please. As you can see here, there were the included studies contained adult and pediatric or mixed population. The MRD time point differed between induction or during consolidation or after consolidation. There were various MRD detection methods used in these publications. And also the AML subtypes contained either core binding factors or non-core binding factors. And also the specimens that were used for assessing MRD differed between bone marrow or peripheral blood, but most of them used bone marrow. And here you see, to make a long story short, the outcome of this meta-analysis showing a really good association of MRD with survival outcomes in those patients with acute myeloid leukemia. On the left-hand side, you see the overall survival. 5-year overall survival for the MRD negative was 68% against 34% for the MRD positive, and the same accounts for disease-free survival. A nice discrimination between MRD positive and MRD negative patients. So really showing the prognostic value of MRD. And in this forest plot, you can see that for all subgroups that were analyzed, you can see they were better for the MRD negative patient, except for cytogenetics, but that is not so strange because it is a really insensitive method. This is disease-free survival, and the same accounts for overall survival. Also here, only cytogenetics was not better for the MRD positive patient, but again, a very insensitive method. So the conclusion, next slide, of this study is that that achieving MRD negative is absolutely associated with a superior disease-free survival and overall survival in AML, and it appears to be consistent across all age groups, AML subtypes, the timing of MRD assessment, the specimen source, and the MRD detection methods, except for the cytogenetics. And these results support MRD status as an endpoint that may allow for the accelerated evaluation of novel therapies in acute myeloid leukemia. I have to underline that this is showing the prognostic value of MRD and certainly not the predictive value. FDA is also very interested, and EMA also, in using MRD as a surrogate endpoint for survival, especially to accelerate drug testing and approval. And there are now examples of hematological diseases in which MRD is already accepted by the regulatory authorities as a surrogate endpoint, and FDA produced a guidance document for making use of MRD. I will not go into detail, but there are some factors that are important. So it has to be biological plausible that the biomarker could serve as a surrogate endpoint. Prognostic value has to be shown already, and that has been shown, I think, by all published studies up till now, and certainly underscored by the meta-analysis that I showed you in the previous slides. But probably, and not probably, but we surely need other analysis to show the predictive value, and that was the starting point of a new project in the AML Working Party. We started a new project in the Working Package 2 AML, and formulated the aim of this study to assess MRD after 2 cycles of chemotherapy as a potential surrogate endpoint for survival. And we will conduct a prospectively planned pooled analysis of individual patient data, and that's different from all the published data until now because they made use of trial data and not of individual patient data. And we will have to use randomized controlled trials because that's the demand of the regulatory authority, and it will be in the first-line therapy in acute myeloid leukemia. And the selection of trial will consist of prospective multi-center randomized trials in patients with previously untreated AML with a sufficient sample size. And because studies of intensively treated patients collect the most thorough data sets, the analysis will be restricted to this population. So not for the unfit population, but for the fit patients that can get intensive treatment. So the surrogate endpoint definition is depicted here, and we need difficult and for me very difficult to understand statistical analysis. And this is based on a study that has been published in JCO a couple of years ago in follicular lymphoma, in which the PFS was used as a biomarker for a surrogacy. And they make use of 2 statistical methods that are depicted in these sentences. I will not go into detail, but it underlines that we need a good statistical team to analyze the individual patient data. And this is what we are currently have. So we have already composed a kind of core group. So Hartmut Deuner, Brian Huntley, Lars Bühninger, and myself, who are lead participants of WPT AML of Harmony. And they will be extended with the representatives of major AML trial groups in Europe. And you can see here the list of all those major AML trial groups that already agreed on this proposal and will participate. So this is AMLSG, led by, represented by Michael Heuser and Constance Deuner, the NCRI by Sylvie Freeman and Richard Dillon, HOVON, Jacqueline Close and Peter Falk. GEMEMA will participate. Adriana De Vendetti and Francisco Bucasani will participate. The SAL will participate with Christian Rullik and Christian Tiede. Alpha have already also committed to this protocol, so Hervé Dombray and Christian Perdom. Philo from...
Hola, ¿me puede decir cuál es el teléfono de admisión, por favor? Sí.
Philo have accepted to participate, and Setlam is currently discussing it. The Polish AML Working Party will participate. We have asked various pharmaceutical companies participating in Harmony whether they are interested to participate in this project. They have not confirmed their participation currently. They are not so important for data delivery because they usually do not have much data on intensively treated patients, except for Novartis with the Verify study. But they are really important for negotiations with the regulatory authorities and also giving their input in this project. And then we need, as I alluded already in the previous slide, a very dedicated statistical team to perform the individual patient data analysis necessary for proving that MRD is a real surrogacy for outcome. So the work plan is here. We have to determine a team of PIs for this study who are really responsible, and that should be one clinician, one MRD technology man, and a statistician. And they should be responsible for developing an eCRF, which is really important, and they should are responsible also for the progress and monitoring and preparing a draft proposal. Making a real statistical analysis plan is a step that is necessary, and developing the eCRF, the molecular data that we already have in place can now be used for extending the database for gene-gene interaction and should then be applied for overlapping data. So that's also, that's partly in place, and we have to prepare a budget proposal. So I've shown you that presence of MRD after treatment is a bad sign. It's prognostic for outcome, but we have still not proven that it's predictive for outcome. If MRD would be accepted as a surrogate marker for survival, it would be important for drug approval. And I think this would be a really good and new outcome measurement that really fits in the aims of Harmony. And the huge data set have to be collected within Harmony to perform the meta-analysis to establish this surrogate marker. And I showed you already that many major AML trial groups in Europe will work together to achieve this important task. So there's a lot of work that has to be done. Our next step will be that we write an official proposal for Harmony. Hopefully they will accept it. I can't imagine that they won't, but I think this is one of the things that we should do. And that's what I want to say, and thank you for your attention.
Thank you very much, Gert, for this very exciting talk and project. And I think everyone will agree that this is a project which we definitely should pursue within Harmony. Are there questions from the audience? So there's one comment from Guillermo, who liked the presentation also very much. And he said that this perhaps also could be a template for the other disease groups. I mean, there's, for example, myeloma, where of course minimal residual disease as a surrogate might also be important, or other other disease group. I mean, from CML, we of course definitely know it.
Maybe one additional remark that I forgot, Lars, and that's that part of the data is valuable. There are already studies prospectively done with MRD assessment, and but there are a lot of studies ongoing currently, and therefore it's really important that this proposal will not only be done in Harmony but also in Harmony Plus.
Mm-hmm. Okay. Thanks for the additional comment. Yeah, I think if there is no additional questions from the audience, I think, yeah, once again, thank you very much. Very good project, and we are all looking forward to it. And I'm now handing over to my co-chair of this session, to Farzad Ali, who will announce the next speaker.
Sure, thank you very much, Lars. So it's a privilege to be moderating the session with Lars, and it's also a privilege to be among you all. Pfizer, as you may know, came into Harmony last year, and we are very excited to be taking part actively in Harmony and Harmony Plus. So if I'm not mistaken, it's not Professor Barbieri who's presenting now, it's Professor Pagano, right? So yes. I will, okay, perfect. So just a brief introduction for Professor Pagano. Professor Pagano is currently the Associate Professor of Hematology at the Catholic University of Sacred Heart in Rome, and serves as an attending physician on the Division of Hematology Policlinico Gemelli, Roma, Italy. And he is the head of the unit for the treatment of hematologic malignancies in elderly patients. Professor Pagano is a member of numerous academic societies, and I believe most of you know him pretty well. And he has co-authored or authored over 300 original manuscripts. So it's with a lot of pleasure that we have Professor Pagano presenting to us the epidemiology of COVID-19 for patients with hematologic malignancies. So please, I will hand it over to you.
Thank you. Good evening, everybody. Thanks for the invitation and for the opportunity to introduce you our study. As already said previously by Guillermo, our survey is promoted by Infectious Disease Working Party of European Hematology Association. It is focused on the epidemiological analysis of COVID-19 in infection in hematological malignancy patients. As for the background, all you know that COVID-19 infection is a clinical emergency that is affecting everyone in Europe and not European countries. Patients are suffering from hematological malignancy are certainly those at highest risk of morbidity and mortality, both for the immune status linked to the underlying malignancy and for the treatment that they must receive. And in the last few months, various papers have been published. Apart the study by Passamonti and which involved all the Italian Society of Hematology and that collected more than 500 cases. All the others are on limited series and on specific pathologies, in example, acute myeloid leukemia, chronic lymphocytic leukemia. So we have no a correct idea of this infection in a large population. So the aims of our survey is to collect information on this infection in all the hematological malignancy patients on a large number, involving all the European centers of hematology, and in order to have a better and clear idea on the outcome of our patients. Regarding inclusion criteria and exclusion criteria, take note that this is a multicentric retrospective prospective study because we want to collect data from March 2020 until December 2020 for the moment, and is a not interventional study, but is only observational. As an inclusion criteria, we want include only adult patients, patients aged 18 years old, and with an active hematological malignancy at any stage or status. And also all patients, in order to be included, must be positive to the SARS test. Patients with non-malignant hematological disease, not with a test negative, or patients off therapy from more than 5 years from the hematological malignancy are, of course, excluded from our survey. Our objective, the primitive objective, is to assess epidemiology outcome of patients with hematological malignancy infected by COVID-19 disease. Of course, from epidemiology, we means a registry of all cases. We cannot have the information regarding all the cases, because there is a problem to have all the data regarding all the patients with the entire hematological patients cohort or on the general population. So we cannot have the concrete incidence of these cases among all the other population. And as a secondary objective, we want to estimate the prevalence and the type of COVID-19 disease in our patients: symptomatic, asymptomatic, a severe disease, how many patients needs of the admission in intensive care units, how are the pre-existing comorbidities that they presented. We want to evaluate, above all, the mortality within the 30 days from diagnosis, and to estimate the rate of overall case fatality. And we want to know also the distribution in Europe of the disease, and stratify the patients for on, off therapy, for kind of therapy, chemotherapy, immunotherapy, cell therapy, stem cell transplant. Of course, we are hematologists, not intensivists or ID specialists. So we not want to know how is the kind of treatment for these patients, because usually in my experience, these patients are not treated from hematologists, but when they present COVID-19 infection, they were followed in another units. Until now, we have the partnership and the endorsement of a lot of scientific group, apart the infectious disease group of IHA, also of the group of aging and hematology. We have the support of a supportive treatment group of a Croatian cooperative group for hematological disease, from the SAFEM, that is the Italian group of survey for infection in patients with hematological disease, from the Danish National Registry of COVID-19, and from Italian Society of Hematology. But all members of HAA will be invited to participate. I want to give you just a few data that we already have collected. Take note that this study started, the collection started at the end of August. So in 1 month and a half, we already collected 355 cases among 57 centers in all Europe, as you can see here. In the arrow for Croatia and France, is indicated that 1 center for 12 centers or for 6 centers. So there is a good representance for these two countries. And I want to also say to you that between 13 October, when I prepared this picture, and today, we had more 50 cases again. So we now have 400 cases already registered in our database. And when the ethical committee of the local ethical committee of the centers that participate give their approval, already 250 cases were obtained. So we will arrive at present to 600 already in our database. And in this slide, I show you how are the distribution of the cases among these 355 patients that now we have. As you can read, the majority of them are myeloma or non-Hodgkin lymphoma, as expected. And it's interesting to see on the last column the mortality rate. The mortality rate, unfortunately, is dramatically high. And among 35% in acute myeloid leukemia, you please see where there is a lot of patients. Chronic lymphocytic leukemia, acute myeloid leukemia, multiple myeloma. It is near 35% for all these patients. Of course, we need more data with more patients in order to have a correct idea of all the situation in all the patients. And the final. We give no limits to the case that we can be enrolled. We hope to arrive to 1,200 patients at least. And I think, okay, considering that the situation of the problem of COVID-19 that is present and is very important, as at the beginning in March, we want to continue our survey until June 2021. So we will have data retrospective and prospective. At January, we did the first, we will perform the first stop, and a first report will be produced and submitted with the preliminary results on the data that we collected. And we are evaluating also the opportunity to a longer-term study on this population, above all to analyze the compliance in patients who recovered from infection with the therapeutic program of hematological malignancy. How many of these patients that are scheduled to perform a transplant after can perform this one? I invite all to participate to our study because I think that more data we have, more information will be for everybody for a better know of this one. Thank you.
Thank you very much, Professor Pagano, for what appears to be a very pertinent and relevant current research. I don't believe there were any questions. There was a general question around presentations, which Ellen kindly responded that they will be available end of day Monday. Are there any other questions for Professor Pagano? If not, we will move on to the next presentation.
Yeah, thank you very much also from my side for this excellent presentation excellent presentation and proposal for a very important research project, I think, which also fits very well into Harmony. And speaking of COVID and the importance also to better understand the infection, also in taking care of our patients, I'm also happy to announce the next speaker, who will be Tiziano Barbui, one of the world leading experts on myeloproliferative disorders, which are now also part of Harmony. He is a hematology professor at Bergamo, has chaired the Italian Society of Hematology, is part of the GMEMA group. I think all of you know him, and I'm happy to hear about your proposal on COVID and myeloproliferative disease. Professor Barbui.
So thank you very much for giving us the opportunity to present the results of our study on MPN and COVID-19. We addressed three questions for this study. The first was whether MPN patients are really at higher risk of death for COVID-19 in comparison with the general population. The second question we addressed was whether cytoreductive drugs should be adjusted during the COVID infection. And the third, whether thrombosis and bleeding could constitute a major concern in patients with COVID. You know that thrombosis and bleeding are part of the natural history of the MPN. So this question could be very relevant. We launched this study we called MPN COVID, and the design is a multi-center retrospective observational study. We, as a foundation for clinical research, are the promoter of this study on behalf of European Leukemia Net. But I have to say that this study was endorsed by EHA and Harmony Alliance as well. Very, very glad to present this data on behalf of these societies. As a chairman of the Working Package 9 of ELN, I am the scientific coordinator of this study, to which 38 centers participated from Spain, Italy, Germany, France, UK, and Poland. The registration of these patients started in February 15 to June 2020, and the analysis were performed in July this year. Patients were diagnosed according to WHO 2016. You see here the 4 different phenotypes of the disease: essential thrombocythemia, polycythemia, myelofibrosis, prefibrotic myelofibrosis. From Italy and Spain, these 2 countries were the major contributors of this series of patients. Remember that we collected these patients during the first wave of the pandemic, where Italy and Spain were particularly in the eye of the cyclone in this period of time. But we have also other centers, as I said, contributing for some patients in the 4 phenotypes. Of these patients, every patient had also the mutation status, the driver mutation, JAK2, CALR, MPL, and we had to not consider 3 patients who presented with acute leukemia as evolution of this disease. So at the end, this analysis pertains to 175 patients. Therapy during COVID at that time were drugs such as hydroxychloroquine, you see 60%, antiviral, 34%, experimental, and antithrombotic drugs were given mainly low molecular weight heparin that accounted for almost all patients who had treated with antithrombotic drugs. Interesting to know is that these patients were treated in regular ward or at home, or were transferred to intensive care unit in about 20% of these patients. So we had three allocation of our patient at home, regular ward, or in intensive care units. So the first question was the mortality. You see here that overall mortality was about 28%. And you see here that 25% of patients died in the first weeks after the admission to the hospital. And then the curve reached a plateau. So the causes of death were very heterogeneous, but I want to point out that almost half of these patients died whether they had myelofibrosis. So myelofibrosis appear to be the most vulnerable disease in this cohort of patients. 48% died, especially in the first days after hospital admission. If you compare this mortality with the mortality of the general population, we found that this is a mortality 5-6-fold higher in ET, PV, and pre-PMF, and is at 10-11-fold higher when you consider only MF patients. The second question regarded whether the cytoreductive therapy should be adjusted and what was the impact of this cytoreductive therapy on mortality. These were the drugs of our patients used during the last follow-up, pre-COVID. You see two drugs were majorly represented, especially hydroxyurea was used in ET and PV in about 60% of patients. And the second drug most used was ruxolitinib. In ET, in some patients, then in PV and in myelofibrosis, particularly 60% of patients in myelofibrosis, in the 60 patients with myelofibrosis, were treated with ruxolitinib. In multivariable analysis for mortality, what we found is that age was important, sex, male, respiratory support, ICU admission, but what we found that was very interesting, that ruxolitinib discontinuation was independently associated with the risk of death. You see here the odds ratio was about 9, but the interval coefficient was particularly elevated. And the p-value, however, was very significant. So discontinuation of ruxolitinib was the major determinant to answer the question on the role of cytoreductive drug for mortality. And this is well represented in this Kaplan-Meier. This Kaplan-Meier is this curve, the highest curve, regards a patient treated with hydroxyurea mainly. And the red curve regards patients treated also during the COVID disease with ruxolitinib. But this was the survival in green of patients who discontinued ruxolitinib. Virtually almost all patients died after 60 days if they discontinued abruptly ruxolitinib after COVID disease. You see here the slope of this curve. In 10 days, you get the most frequent mortality, and most 70% die in the first 10 days. Since this data were generated from data from univariate and multivariable analysis, to be more solid with this data, we did also a propensity score matching analysis. Of course, the number of patients were reduced to have a perfect matching with the population, but we repeated exactly the observation. As you see here, in 2 weeks, all patients died if they discontinued ruxolitinib. So the third question was about thrombosis and bleeding. What we observe in this competing cumulative risk, the incidence of thrombosis. You know that the thrombosis in this disease is 2-4-fold higher outside the COVID disease, higher than the general population. So the question was whether the incidence of thrombosis would have been higher if this patient got COVID. Really, we observed that the incidence, the cumulative incidence of thrombosis in this competing risk cumulative analysis, by taking death as competing events, was about 8.5%. Concerning bleeding was about 5%. These rates are not so different from those registered in non-cancer or cancer patients. Multivariable analysis was done only for thrombosis because the low number of bleeding events precluded multivariable or other analysis, statistical analysis. What we found is that ET was the most susceptible disease, having a patient having a high risk in multivariable analysis, independently from neutrophil lymphocyte ratio that was also independently associated with thrombosis. And of course, also ICU transfer was associated independently with thrombosis. What we found a very interesting finding regards ACE and ARBs inhibitors. These were protective against the thrombosis. And as you know, there is a biological plausibility to protect the endothelium if you give ACE inhibitors, because people think that by blocking the receptor of the virus, you can reduce the property, the prothrombotic property of an endothelium that is attacked by the virus. So, in summary, mortality in MPN is elevated. Among the 4 clinical phenotypes, the risk of death was 48% in myelofibrosis. Very careful then when a patient presents with myelofibrosis. Unexpected results is the ruxolitinib, and overall mortality that is not due so much to the drug exposure, but is due to its rapid discontinuation at the COVID diagnosis. Thrombosis and bleeding is not higher than in general population. There is a protective role of ACE inhibitors. Further analysis, however, we need to study the effectiveness and safety of low molecular weight heparin in this setting. The last one, that is an important slide, is the enlargement. I have to acknowledge the European centers that enthusiastically collaborate with us. All my co-workers, these 2 people, these 2 young ladies are the biostatistician. This is Arianna Masciulli. She is the project manager for us. And of course, I have to thank very much the endorsement of EHA and Harmony, and especially European Leukemia Net. And you for listening. Thank you.
Tiziano, thank you very much for this very interesting study. And I think you raised a very important point that it is important to continue therapy for the patients during COVID and that we should not stop it. And you also have, of course, a very nice working environment. Congratulations. I think because of the shortness of time, I think we have to continue. I'm sorry. And I hand over again to Fasad Ali for the next talk. Thanks again.
Thank you. Thank you very much, Lars. Great talk. So the next talk is by Dr. Santiago Redondo. It's one of the FPIA submissions, and it's a validation of the findings of the machine learning analysis of Optum in polycythemia vera patients. So with Dr. Redondo is an hematologist within Novartis. He has completed his European PhD in pharmacology. He's a physician, and he was a clinical hematologist at the Hospital of Avila, an emergency doctor in Madrid. Without further ado, due to time, I will just hand it over to Dr. Redondo.
Good afternoon. First of all, I would like to thank the Harmony Consortium for giving me the opportunity to present this collaboration. And this is a collaboration by the Novartis Oncology Region Europe, and it is the external validation of a machine learning approach performed in a database. We can see that machine learning techniques involve the massive analysis of huge amount of data, and these approaches can generate new variables and hypotheses that didn't receive attention so far. Previously, we analyzed by machine learning a database from the USA, the Optum database, which is a massive clinical database, and we tried to find if in patients of polycythemia vera, we could predict the coming of hydroxyurea resistance or thrombosis, which are 2 very important clinical problems. These results were presented at last edition of ASH. And just as an introduction and as a fast track of the most important results, we found that the phlebotomy dependency and the increase in red cell distribution width at baseline before giving hydroxyurea to the patient could predict the coming of hydroxyurea resistance within the next 3 months. In addition, for the patients with PV that were treated with hydroxyurea for 6 months, we could predict the coming of thrombosis within the next 12 months. And it was a difference with the patients without or with a previous history of thromboembolic event. And just to be brief, the neutrophil lymphocyte ratio and the decrease of lymphocyte, also the red cell distribution width and the number of platelets could predict the coming of thrombosis. And these items and this assessment are not registered in the majority of studies so far. In addition, we made a retrospective analysis of the patients with hydroxyurea who developed hydroxyurea resistance and switched to ruxolitinib versus the one who continued on hydroxyurea, and we found a trend towards lower thrombosis in the patients who switched to ruxolitinib. Of course, these are hypotheses posted by machine learning and should be validated in another databases. And the ideal database could be a massive database with a large amount of data. And if the amount of data is strong enough, we could make conventional statistics just to make a strong statistical potency, just to see if this prediction could, if these items could predict the coming of hydroxyurea resistance or thrombosis. And in addition, we could compare in a receiver operator curve this putative novel risk factor for resistance or thrombosis versus the traditional ones. And in addition, we could combine them all in order to build a statistical prediction in order to, given a single patient, before giving the first dose of hydroxyurea, to predict if the patient needs a high probability to develop hydroxyurea resistance in a short amount of time, in the next 3 months. And in addition, if a patient is given hydroxyurea, in a patient of polycythemia vera, of course, is given hydroxyurea for 6 months, to develop a prediction that this patient is at high risk to develop thrombosis within the next 12 months. And in this manner, this patient could benefit from a strict clinical surveillance in order to try to avoid these thrombotic events. Of course, we think that the Harmony database that now will incorporate MPN data could be a very interesting database in order to validate this hypothesis. We would like to just to reproduce for patients who have gone hydroxyurea resistant, if we have the baseline phlebotomy dependence on red cell distribution with three months before, and see if this could be predicted by these items. And a similar approach could be done for the patients who have been treated for hydroxyurea for 6 months, and then thrombosis is assessed within the next 12 months. This could be a retrospective or even an ambispective with a continuous data acquisition could be done. The final approach, methodology, and protocol is still under construction, and I think it is a nice opportunity to try to validate putative novel risk factors for hydroxyurea resistance and thrombosis in PV patients, and using the Harmony big data in order to validate these questions that could be of interest from the clinical point of view. just a first introduction of this project. Thank you very much for your attention.
Thank you very much, Dr. Redondo. Quite exciting. I believe we don't have any questions. Then for the sake of time, we're gonna, Lars, I'll hand it back to you for the last presentation. Thank you very much, Dr. Redondo.
Yeah, thanks also again from my side for the presentation and the project. We're looking forward to it. And now I'm happy to have be able to announce Andreas Hochhaus. So we reserved the best for the last, more or less, who is going to talk to us about the future of CML in Harmony Plus. And I think you all know Andreas Hochhaus very well, who is the medical director of the University Hospital in Jena and yeah, one of the CML experts in Europe. Andreas, the stage is yours.
Thank you very much for these nice words, and thank you all for including CML, for having the chance to include CML into Harmony Plus. We are very happy to participate, and I want to show you on a few examples what we are able to contribute here and what we did in the past. And we were not included in the initial Harmony because we had a good and a well-functioning network in the context of ELN and the work package 4 in ELN. And as you know, of course, BCR-ABL is the hallmark of CML, and therefore we want to be a little bit separated from the BCR-ABL negative MPNs because the questions may be different here, and it's good to have two columns here for CML and for MPN. And we know that during the course of the disease, genetically defined subclones evolve. We see BCR-ABL reactivation, that means BCR-ABL mutations, for instance, but we see also BCR-ABL independent resistance. And this BCR-ABL independent resistance is, of course, a target for next generation sequencing studies to demonstrate how these different subclones in the course of CML, dependent on treatment response, but maybe also independent of treatment response, evolve. Since BCR-ABL positive stem cells persist, we know that in all patients, even in the patients who are MRD negative by very sensitive PCR strategies, RT-PCR strategies, they persist and due to BCR-ABL independent signals, we know examples from BCL2 activation. We know studies now from BTK activations. That means it's a very interesting field. BCR-ABL independent pathways which drive or which keep these stem cells alive, and maybe a good target for combination treatments to achieve treatment-free remission, stable treatment-free remission, these patients. Therefore, I think the current approach is to establish NGS strategies which may reveal the hierarchy of these genomic events which are responsive for persistence and resistance, which are two different aspects of CML treatment. And we aim to use clinical data and collect additional molecular data to address these questions of resistance and treatment-free remission optimization. So, and what we did in the past was a big study, a big collection of patient data, and Markus Fuhrmann and Michael Lausecker, who were not able to make it today, asked me to present their data here from the UTOS collaboration. The UTOS was a private-public partnership with Novartis within the European Leukemia Net, and we had three sections here. We had a section for patients, and we collected the data from these patients in prospective controlled clinical trials. We had an out-study section and a separate population-based section with very well-defined areas in Europe. We did a prospective collection of data in 20 European countries here. In total, we have a database on 6,602 patients with treatment data and with clinical response data. So, and a few examples what Markus and Michael did with this data. First is to establish the UTOS score. The UTOS score was established to demonstrate or to, because the tyrosine kinase inhibitors have a different impact on the prognosis of patients, and therefore it was important to establish after Sokal and Hasford a new score. And the problem was with this score that the main endpoint, namely survival, was not available in the beginning because only a few patients died in CML. And therefore, we used the achievement of complete cytogenetic remission here, and we established a score, a very simple score, out of basophils and spleen size to demonstrate the chance to achieve a complete cytogenetic remission within 18 months. These are the curves here. You see the separation of the complete cytogenetic remission rate after 18 months, left for progression-free survival, and on the right on the cumulative incidence of complete cytogenetic remission, and you see the clear separation. And this has been validated. Next slide. On this has been validated in an independent course, but then later we were able, with more data, with more clinical data available, that means more patients have died, and we knew the cause of death, and we were in particular interested in the patients who died from CML. Of course, not from other diseases, from older age and whatever. We're interested specifically on the CML as a cause of death. And this enabled us to calculate a new score now with 4 parameters: age, spleen size, blasts, and platelet count. And this score has now the endpoint of CML-specific survival. You see the separation here. Of course, the risk of death is low. Therefore, the curves are very low on the bottom, but there is a statistically significant separation of low, intermediate, and high-risk patients in the learning sample and also in the validation sample. And then, in addition, we have a new blast crisis registry. Blast crisis is still an important problem in CML. We know that additional genetic aberrations, so-called ACAs, additional cytogenetic aberrations of high risk, herald blast crisis. Therefore, it's important to connect the next generation sequencing data with the blast crisis appearance. And we started with the blast crisis registry. Now we have currently 97 patients from 7 European countries in this registry. And we have, of course, new prospective studies from the work package 4 in the European Leukemia Net from all participants. As examples, I show you here two, the TIGER trial in Germany and the Fascination trial. These are 2 prospective trials which are recruiting or have recruited and are available for a database. And of course, there are trials from other countries as well, from France, Spain, Italy, and Great Britain and others. But we did not ask these PIs from these trials so far because we wanted to propose what we want to do first here in this meeting. So and now the scientific part, the molecular part, what we want to do, and I already mentioned the clonal dynamics in CML. We know that additional cytogenetic aberrations have prognostic impact. We know the impact of pH negative clones. That means outside of the BCR-ABL positive cells, we see molecular aberrations. And we know that BCR-ABL may be acquired and produce CML in patients who have already CHIPs. That means the older population with A6L1 or DNMT3A in the hematopoiesis, which acquires BCR-ABL, but we don't know whether this is by accident or whether this has a system, and we need to establish this with more patients. We have a clonal selection of BCR-ABL1 mutations that's already known, but we know a new site now for the new drug, which is now available, the asciminib, which has an allosteric binding site. And on this allosteric binding sites, we have new mutations which were not known before. And therefore, we need to sequence 2 different sites. And I already mentioned the persistence of BCR-ABL positive cells in deep or complete molecular remission. And it's of interest to check these specific cells on a single cell level for additional molecular aberrations. So this is an example here. This is a summary which has been published by Sue Branford as a result of an international collaboration, just a collection of different sequencing data. You see that we see additional chromosomal aberrations at diagnosis in red, mainly A6L1, but also DNMT3A or RUNX. We see BCR-ABL kinase mutations in accelerated phase and blast crisis very frequently. And we see also additional aberrations in accelerated phase and blast crisis in blue. That means that's frequent. This is not unusual, but has not been collected systematically so far. One of the smaller examples, but I think a rather interesting example, is the external induction of such aberrations. And one example was the Chernobyl disaster, and we had the chance in collaboration with our colleagues in the Ukraine, with Irina Diagil, to sequence 20 CML patients who were in contact with the Chernobyl disaster, either living in the environment or working in the plant. And 13 of these 20, that means a higher proportion than usual, had additional molecular aberrations, 7 of those showing more than 1 mutations. That means we know that external environmental factors may induce such additional clones as well. So, and we summarized our proposal, what we want to do in this paper, which has been published in Leukemia last year by Sue Branford and colleagues. And we created within the International CML Foundation a sequencing group. And we were still thinking about the way to bring this data together. But since you announced Harmony Plus last year, this has been not delayed, it has been postponed until now, because of course all these groups here on the top left, the European Leukemia Network Package 4, the UTOS Data Center in Munich, and the UTOS sequencing labs, and all the labs in the international CML foundations are interested to contribute sequencing data with clinical annotations of these patients. And it would be nice to have all these groups and individual labs here connected to Harmony Plus. So this is a roadmap. We want to transfer the registry data, which I mentioned, to have the clinical background from more than 6,000 patients. We want to complete new registry, like the blast crisis registry, for instance. And we want, that's the main part of this, our project, to collect annotated sequencing data within BCR-ABL and outside of BCR-ABL, and of course correlate this clinical data with the outcome. And if all these people who expressed interest will participate in this trial, we will, in a short period of time, collect a lot of data here. And I think it's very nice that Harmony Plus has now offered the chance to include CML which is a BCR-ABL-driven disease, but all which is outside of BCR-ABL might be even more interesting than BCR-ABL itself. Thank you very much.
Yeah, thank you very much for a very nice project that fits perfectly in Harmony Plus, and that we already have such outstanding data collections available that can go into Harmony Plus will definitely make the CML project a success. Yeah, so I think while I would love to ask many questions, unfortunately the time is almost over. And with that, I think I will hand over again to Jesus, and thanks again, Andreas, for this project. And I'm looking forward to hear more about it in the next meetings.
Okay, thank you, Lars. Thank you, Farsad. And I also want really to give a very big thanks to all the 5 speakers. We have 5 really exciting projects, outstanding projects, let's say, because it is quite important that we don't have to choose one of these projects. We can have all the projects in the frame of Harmony. We can provide from Harmony and Harmony Plus the infrastructure to collect the data coming from the NPM, from the COVID-19, from the P-beta, and from the CML patients. And this is quite important. And it's also quite important to enlarge Harmony projects with these MRT projects regarding AML. That is also an outstanding project, covering and join and putting together different working cooperative groups across Europe and even across the world. So it is quite important to have these new ideas coming to Harmony. It means that we have a lot of work still to do, but also we have very good ideas now coming, and these ideas will generate new results and outstanding results in all the frame in covering all the hematological malignancies. So I have to close here the meeting. It has been 3 hours yesterday, 3 hours and a half yesterday, 3 hours today, full of discussions, interactions between us. Sometimes I think, at least me, I missed a little bit the interaction with the other one due to the pandemic, due to these restrictions that we have. Maybe we can, in some way, we can change this by using the time of the next General Assembly on February. And we will also organize focus webinars in myeloma, in ALL, at the time that we will start to have some data in order to discuss these topics point by point. Thank you so much for attending the meeting. Thank you so much to the speakers. I know that it was a very short time for some of you to be invited to this meeting, but it is the arrangement, the last arrangement of the meetings were performed in the last, let's say, 2 or 3 weeks. But you were available at any time, and I'm quite sure that we will keep in touch, and we will take a lot of interaction from now. Thank you so much. Harmony is alive even in these pandemic times. And at that time, we have to work. A lot of work is coming, and please, we will really thank all your feedback regarding these sustainability ideas that Dr. Muller also pointed in the first meeting, in the first session. Thank you so much.